Hello, my name is Leena. I'm a Bio 5A SI, and today we're going to go over nucleic acids and cell membranes. So nucleic acids are the fourth macromolecule that we're going to learn about, and their monomer is called the nucleotide. So we use nucleic acids for genetic storage, like in DNA, energy in ATP, and in coenzyme activities, which we will learn more about in cellular respiration and photosynthesis. So nucleotides have three main components. You have the pentose sugar, you have the nitrogenous base, and you have the phosphate group. And I drew the Lewis structure right here, just so you can see it better. So the main difference between DNA and RNA is, if you can see, I've labeled the carbons, 1', 2', 3', 4', 5', is that in DNA, deoxyribonucleic acid, there is no hydroxyl group on your 2' carbon. But in RNA, there is. So if I just drew a hydroxyl group here, that would make this an RNA monomer. So there are two types of bonds that you could see in nucleotides. We'll talk about the ester bond first. This is a dehydration reaction that basically connects a nucleoside, which is just the pentose sugar and the nitrogenous base, with the phosphate group. So you could pause here and see. And this also connects the multiple phosphate groups that you could have on one nucleotide, like in ATP. And the next bond is the phosphodiester bond, which is what connects the nucleotides together and basically makes the polymer, which is like DNA or RNA. So by the red arrow, you can see where I've labeled where the dehydration reaction occurs. And so the phosphodiester bond, once you have this polymer, you have two ends of your strand. You have the 5' end with the phosphate, and you have the 3' end with your hydroxyl. And this is how you can basically tell the difference on a strand. So we know that DNA is double-stranded. So DNA is now anti-parallel. It has a 5' end to a 3' end, but the other strand goes the opposite direction. So the 5' end of that one is with the 3' end of the other one, and like vice versa. So this gives the strand directionality. You can just tell the difference between what side of the strand you are on based on what's on the end. So let's talk about the nitrogenous bases. So you have like two types. You have purines and you have pyrimidines. Purines have double rings, and they are adenine and guanine, and pyrimidines are cytosine, uracil, and thymine. So the main difference is that purines have double rings and pyrimidines are just single rings. So purines hydrogen bond with pyrimidines, so adenine goes with thymine or uracil. Uracil is just for RNA, thymine is for DNA. And then guanine hydrogen bonds to cytosine. So let's start talking about the cell. So the cell is the smallest unit that meets the criteria for life, and it has to have 6 requirements. It needs to acquire and use materials for energy. It needs to maintain organization, which we'll learn about through like organelles. It needs to respond to external stimuli, which we'll learn about in like cell transduction. And it needs to grow, which you can see in like meiosis, mitosis, and also reproduce and evolve, which you can learn later in like the Bio 5B. So the cell theory basically states that every organism is made of cells. Cells are the basic unit of structure and organization, and all cells arise from pre-existing cells. So let's start talking about the plasma membrane. The plasma membrane has 3 main components. It has the phospholipids, which we learned about in the lipid section. It has embedded proteins, and it has the cytoskeleton attachments. I don't know why that word is so hard to say. But yeah, so the phospholipids basically makes the membrane semi-permeable, which we'll learn about more in a minute. So the cell membrane can be fluid depending on the certain situations that could happen. So depending on temperature, if you increase the temperature, there's going to be more kinetic energy, things are going to move around faster, so the membrane will be more fluid. But if you decrease the temperature, it's going to slow down and make it less fluid. And saturated versus unsaturated cells, this depends on your cis-double bonds. So cis-double bonds make your membrane more fluid because basically you have kinks, and that gives it more room for things to move around. And cholesterol. Cholesterol acts like a sort of buffer where it minimizes the effects of temperature on fluidity. So if you have a really high temperature, your membrane is really, really, really fluid. Adding cholesterol will make it a little less fluid so it doesn't fall apart, and vice versa. So let's talk about the fluid mosaic model. Basically, fluid mosaic model says that mosaic of protein molecules can move around in the fluid lipid bilayer. So you have three types of proteins. You have transmembrane, peripheral, and integral, depending on whether they completely cross the membrane, or they're just on the edge, or they only go like halfway. So these proteins that can be embedded in your lipid bilayer can be made up of alpha helices or beta pleated sheets, and the beta pleated sheets are mostly seen in just like bacteria. So let's talk about membrane proteins. Membrane proteins can serve a lot of functions. They can be used for transport, they can be enzymatic, signal transduction, cell-to-cell recognition, intracellular joining, and exoskeleton matrix joining. And we'll learn more about these types of proteins as we go throughout the course. So like we'll learn about enzymatic, signal transduction, and yeah. So when we say the membrane is semi-permeable, it depends on what can actually cross through the membrane on its own and what needs help. So small nonpolar molecules like oxygen don't really need help crossing the membrane, so they can freely move from out and in the cell. But large polar slash charged molecules or ions need a transport protein because they can't cross that hydrophobic layer. And water, if you want to move a large amount of water into or out of the cell, you're going to need an aquaporin. And an aquaporin is basically a transport protein that is mainly used for the transport of water. So these types of transport proteins, there's two types. You have passive and you have active. And we're mostly going to talk about passive today. So passive means that you don't need any energy to move things around because it uses the concentration gradient that naturally happens, that happens spontaneously. So what happens spontaneously is that solutes with a high concentration will try to move to the place that has solutes in a lower concentration to try and even it out. So there's three types of passive transport. You have simple diffusion, which is it just happens on its own, like oxygen just moves through the hydrophobic layer to its like concentration gradient. You have facilitated diffusion, which needs a carrier protein, which is like large charged molecules. And you have osmosis, which is basically the movement of water, which we'll talk about more. So with osmosis comes these three types of situations of tonicity. So depending on the solute concentration in your cell and outside the cell is whether the cell gains or loses water. So in an isotonic environment, the solute concentration inside the cell is equal to the solute concentration outside the cell. So there's no real gradient that the water needs to move in to even it out. In hypertonic, this means that the outside environment has a greater solute concentration than inside the cell. And water wants to even that out. So most of the water that's inside the cell is going to go outside to try and even it out. In a hypotonic environment, the outside environment has a lower solute concentration than inside the cell. So all the water on the outside is going to try and go inside the cell to try and even it out. So you can talk about whether the animal cell is lysed, turgid, or normal, or whether the plant cell is like plasmolyzed, turgid, or flaccid. And that's the main difference. So if you have any questions, feel free to put it in the comments below, and good luck.